Hey guys, welcome back. I'm Kritik, a first year MBBS student in ESIC Medical College and PGIMS of Bangalore. Now, coming to the topic lipogenesis, de novo synthesis, that is cytosolic synthesis, or also called as extra mitochondrial synthesis. It is so called because the process is taking place in cytosol and not in the mitochondria. Now, lipogenesis, when does it occur? It occurs when there is surplus to glucose or when the glucose levels are high, so much that it is more than what is required and it is converted into storage form of fat, that is triglycerols. Now, the major site are primarily liver, mammary gland, and small amount of adipose tissues. The starting materials is acetyl CoA. So, as we know, acetyl CoA is majorly in the mitochondria, so it has to be brought back into the cytosol, and so that the fatty acid synthesis can occur. So, this is brought about by malate oxaloacetate shuttle. Where in mitochondria, acetyl CoA reacts with oxaloacetate in the presence of citrate synthase will form citrate. Why do you need citrate? Is the question, right? So I'll tell you that acetyl CoA cannot cross the mitochondrial barrier directly. So citrate can cross the mitochondrial barrier directly, so that the citrate crosses the barrier and enters the cytosol, wherein it is acted upon by ATP citrate lyase. Remember this, ATP citrate lyase to form back acetyl CoA and oxaloacetate. The acetyl CoA is what we require to form or for the lipogenesis process. So there is requirement of two enzyme systems. First one is acetyl CoA carboxylase system. Second one is fatty acid synthase complex system. So we need to know something about the fatty acid synthase complex system. This fatty acid synthase is a complex enzyme system wherein it is in a dimer form, and it is functional only in the dimer form, and all the enzymes that are derived are from single gene. Okay. To make our job easier, there is a mnemonic. A big shout out to Rakesh Jambulkar sir for making it such simpler. Okay, we'll go with the mnemonic, and the ones which I've written in the caps are the ones which are important for us. Okay, now going with the mnemonic, ATM transaction kit started a refund of estimated dollars, rupees through ACP. I'll repeat it once again. ATM transaction kit started refund of estimated dollars, rupees through ACP. Now we'll go in an order, and the numbers which I've assigned are very important for you to remember it the same way, because it will be used for its structure. Now AT is acetyl transacylase, MT is malonyl transacylase, KS is ketoacyl synthase, KR is ketoacyl reductase, ER is enoyl reductase, D is dehydratase, T is thioesterase. ACP is acyl carrier protein. There is something interesting about acyl carrier protein is that in prokaryotes it is a single entity. In eukaryotes it is along with the fatty acid synthase system. Now coming to its structure, it consists of a condensing unit, reduction unit, releasing unit. The same thing should be written in the opposite manner below. It makes us write the structure easily actually. And then there is two subdivisions. The one which I've drawn now is called as functional subdivision. The horizontal division is called as structural subdivision. And then yeah, now in the order which we have to write from left to right in the above line horizontally is 312 654 ACP 7. The same way in the reverse manner should be written below, 312 654 ACP 7. Now these are the numbers which we have assigned for the synthase fatty acid synthase complex enzyme system. And then to 3 there is attachment of cis and thiol groups. The same way in the opposite side it is attached to ACP. The same way here also. 3 is attached to cis, and then thiol groups is attached to ACP. Now the releasing unit being 7, reduction unit being 654 and ACP, and then condensing unit is 312. Now you are familiar with fatty acid synthase complex system. We'll move to the steps of the reaction. The first one being the rate limiting step, that is acetyl CoA along with the carbon dioxide in the presence of acetyl CoA carboxylase will form malonyl CoA. This reaction is a carboxylation reaction, and carboxylation reaction requires ABC in short. A being ATP, B being biotin, C being carbon dioxide. So ABC is required in carboxylation reaction, and this is the rate limiting step. Now, the basic idea is there is requirement of 8 acetyl CoA. 1 acetyl CoA is directly reacted with 7 malonyl CoA, which is formed from 7 acetyl CoA by the reaction of acetyl CoA carboxylase to form palmitic acid. This is the overview of the reaction. Now, acetyl CoA reacts with cis enzyme thiol in the presence of the first enzyme, that is acetyl transacylase, to form acetyl enzyme and CoA. Malonyl CoA along with ACPSH in the presence of malonyl transacylase will form malonyl ACP. Now, there is 4 important steps further, which is very important, with the first step being the condensation step, wherein the acetyl enzyme and the malonyl ACP both condense to form beta ketoacyl ACP in the presence of beta ketoacyl synthase. Next step being the reduction step, beta ketoacyl ACP is reduced under the influence of ketoacyl reductase to form beta hydroxybutyryl ACP. In this step, there is a production of NADPH2. And the third one being dehydration reaction, again beta hydroxybutyryl ACP in the presence of dehydratase will form enoyl ACP with the liberation of water. Then again reduction, enoyl ACP is reduced to butyryl ACP under the influence of enoyl reductase, which again forms NADPH. Now, these 4 steps, that is condensation, reduction, dehydration and again reduction, will continue for another 6 cycles to form palmitic acid ACP, and this palmitic acid ACP is acted upon by deacylase or thioesterase to remove the ACP, and finally palmitic acid is formed. Now, coming to the regulation, acetyl CoA carboxylase enzyme is regulated, and it is the rate limiting step. It is stimulated by insulin and inhibited by insulin antagonists. Now, coming to the fate of palmitic acid, there is 2 fates that the palmitic acid faces. First one is the chain elongation, second one is the desaturation. And again, under chain elongation, there are 2 types: microsomal chain elongation and mitochondrial chain elongation. And desaturation is a process which requires NADH and molecular oxygen, which converts palmitic acid into palmitoleic acid and stearic acid into oleic acid under the presence of fatty acyl desaturase. That's it. Thank you.